As discussed in my Learning Object 1, Mycobacterium tuberculosis is a rod-shaped bacterium that is found in airborne particles, otherwise known as droplet nuclei. It is an intracellular pathogen that results in infection of tuberculosis. Once infected with this bacterium, the host's immune response includes components of both the innate and adaptive systems. The innate immune system elicits an immediate response that works to prevent microbes from entering sterile sites in the human body, eliminating a potential threat. Its two defenses against pathogens are passive, such as skin and mucus, and active, such as macrophages and complement. The adaptive immune response is not as immediate, but it is more specific and has the ability to recognize a vast amount of non-self molecules. One of the major effectors of adaptive immunity is the antibody. When looking at the innate immune response of Mycobacterium tuberculosis, this bacterium will first attack the lungs' first response immune cell, the macrophage, upon infection, where it will release small bits of unidentified DNA. This is detected by a DNA sensor molecule called cGAS, found in the macrophage. The macrophage responds by releasing type 1 interferons and interleukins, as well as harboring the pathogen and transporting it to draining lymph nodes. Other innate immune cells involved in TB infection are dendritic cells, neutrophils, and natural killer cells. These cells recognize foreign bacteria through pattern recognition receptors, or PRRs. However, they can also be recognized by toll-like receptors, TLRs, nod-like receptors, NLRs, and C-type lectin receptors, CLRs. The host produces multiple signaling cascades via PRRs, which launches processes such as phagocytosis, apoptosis, and inflammasome activation. However, this bacterium has a special ability to evade killing by phagocytosis by preventing the fusion of the phagosome by the lysosome. This inactivates the proper forming of oxidants, therefore causing the phagocytized bacteria to evade killing by the macrophage. This process is mediated by nitrogen intermediates, which are formed due to nitric oxide and superoxide anions. The adaptive immune response of tuberculosis is T cell dependent, involving primarily CD4 and CD8 T cells. The adaptive immune response for this bacterium is characterized by processes such as antigen presentation, co-stimulation, and cytokine production. Antigen presentation occurs by MHC class II molecules presenting mycobacterial proteins to CD4 T cell receptors and MHC class I molecules to the T cell receptor of the CD8 cell. T cells and B cells require co-stimulation, in addition to antigen binding, in order to become activated. Co-stimulation is the danger signal which causes the adaptive immune response to only occur when there is a threat from a pathogen. After presenting peptide antigen to the MHC, which is recognized by the T cell receptor, there is binding of the antigen, which signals co-stimulation. After antigen recognition, B cells work to produce clonal expansion and specific antibody production. Cytokine production of activated macrophages and dendritic cells are important in order to stimulate T lymphocytes. In patients with recurrent tuberculosis infection, genetic mutations have been found in genes encoding an IFN receptor, which is key in receptor signaling of macrophages and dendritic cells. Reduction in the production of pro-inflammatory cytokines can result in a delay in antigen-specific T cell immunity.